All right.
Very appropriate title there in Dr. Gain. We're back and we're recording.
All right.
And go ahead, Todd.
Okay. All right. You can see my screen, so I'll just go ahead and thank you again, Kit, for this invitation. I'm going to keep it pretty informal. Feel free to really appreciate the opportunity to share kind of a story of essentially 5 years of thinking about developing and then building and then releasing a computational platform for synthetic DNA screening. And I'll kind of explain a little bit through the presentation why I picked this title, but it's been certainly a fun journey over the last 5 years with my collaborators, and happy to share this story with all of you here today. And thank you for joining on what is for some of you likely a holiday today. So just go ahead and get started. Okay, so I'll just get started by introducing myself. I'm an assistant professor at Rice University in Houston, Texas. My research focuses mainly, it started off on metagenomics and just comparative genomics of microbial genomes. It has since developed into a strong focus on biosecurity and pathogen detection over the last 10 years or so. And then the project I'll share with you today has been really something I've worked on now for about 5 years. So on the current slide, it's just overview of my lab at Rice University in the Department of Computer Science. Have featured 8 PhD students and a postdoctoral scientist and a software engineer. And the 2 individuals, lab members that have kind of highlighted in this red box are the 2 kind of primary contributors to the software tool I'll kind of describe today, with Advite Balaji, the PhD student who's really been leading the charge over the last couple of years here. So I'll just go ahead and get started. And one last thing, just for those that don't know me, my research areas really kind of sit at the intersection of data structures and algorithm design, software engineering. I like to build software tools that others are able to use and can deploy to laptops, to cloud, et cetera. And really, as I just said, biosecurity, metagenomics, and pathogen detection are kind of 3 broad topics that I focus on. Okay, so I'm going to take you all back to where I started. And it was actually here in Paris, France, where I am currently, where I started my postdoc at the Institut Pasteur, and really thinking of genomes as documents of evolutionary history, and not just any genomes, but microbial genomes specifically. And this is really where it all got started. And this is back in 2008, so about 14 years ago. And I really since my postdoctoral essay, really kind of become fascinated with bacterial genome evolution, and then currently metagenomics. And so specifically, again, my research is focused on finding kind of biologically relevant needles within genomic and metagenomic haystacks. In fact, during my PhD, kind of dreamt of the day where we would have these, just tons and tons of data. At the time, really, while doing my PhD, we really just only had a handful of genomes comparatively for any specific bacteria of interest. But for the purposes of this talk, let me refocus it a little bit to be a little bit more on point. And let's think of genomes as documents of engineering and tinkering. Okay, and this is something this is a newer topic for me, and this is a topic that has emerged since obviously it's been around for a while, but really something I've started to really dig into over the last 5 years. And so thanks to advances in DNA synthesis and genome editing, obviously you all know that we can create and modify DNA sequences to meet specific needs and answer research questions. So this new paradigm I've thought about over the last 5 years. Okay, so we can now accurately read and write DNA. And then the central question underlying this research project I'll describe today is, what about accidental or intentional writing of harmful DNA? So that was really at the core of this IARPA FungiCat program I participated in, and just broadly was a topic of interest to me again. So I'll just kind of take you through some of the software I've developed and some of the questions I've encountered and lessons I've learned over the years. Okay, so let's say lesson number 1. This is all going to be phrased around these lessons, right? So lesson number 1, I came into this project and program really kind of humbled by the challenges that was put forth and kind of the questions that others wanted answered. So I just wanted to start with, certainly I came in this thinking it was going to be a computationally challenging problem. And then I would say on the other side, 5 years later, still appreciating the fact that it is a computationally challenging problem. And not only that, there's kind of five kind of facets to this that make it challenging. And these are not kind of comprehensive, but things that stand out to me. One of which is just the need, if you want to deploy this in a setting, in terms of just thinking about screening intentional or unintentionally created harmful DNA sequences. It needs to be able to scale up to maybe hundreds, thousands of variants simultaneously. We need to think about variable length input sequences. We need to evaluate potential biorisk of novel sequences that we haven't seen before, which is again a major area of research for my lab at Rice. We need to think about predicting biological impact of edits or changes to DNA. And again, we want to really try to reduce the computational cost such that this can be deployed in a wide variety of settings. So again, over the past 5 years, the open source software solution that my lab has developed in collaboration with Signature Science is SeqScreen, and this again will be a software tool that I'll lightly describe throughout this presentation. And again, just to highlight this, really think about DNA sequencing. It must be accessible, affordable, and reliable. In addition to any other considerations, but really thinking SeqScreen was developed through the lens of accessibility, affordability, and reliability. Okay, so kind of hit pause for a second. Why do we care about this problem? Or why is it challenging? Or what are some things that I've thought about for the last 5 years when I first started this project? Well, when I started, it was kind of someone coming to me with these A, B, or C choices of different illegal nucleotides and asking the question: is which one of these or are all of these, quote unquote, harmful to a human host? And I didn't know how to answer that. I obviously, there's a lot of nuance to that. There's a lot of other things I'll discuss throughout this presentation, but really that a simple question seemed like very impossible to answer, right? I don't even know. We could think of different ways we could reason about this to figure this out, but really what it came down to is really it launched several years of just thinking carefully about limitations of computational modeling and prediction when we're talking about very, very short fragments of DNA. So that's really where I started this whole project. So let's look on the other side of this. So ideally, in an ideal world, one could say that first A was maybe a housekeeping fragment, not harmful. Maybe the middle represents some type of toxin motif. This could be maybe related to some toxin of interest. And then the final sequence there is just too short to tell. Maybe in the ideal world, one could think, oh, we could just assign these labels and then screen at 100% accuracy and be done with it. But actually, obviously, with these sequence lengths or even things that are longer, but not much longer than this, and kind of real world, what happens is the first sequence will just get a bunch of ambiguous hits. Potentially, we'll just say it looks like a bunch of other things in the database, and the database is huge. One of the main databases we use, and I'll talk to this later, is UniRef, and which contains 30 million cluster representatives. So this might just hit a large portion of those. Maybe the middle hit, given the length and the characteristics of that sequence, it'll just come back and say there's no significant hits there. And then finally, the last one could be no hits. So from the ideal world of getting very specific information that's potentially actionable, or at least could help inform someone after review, to really no information. So this is kind of the extremes I've been thinking about in terms of how useful is this? How can we make this most useful in a setting where it could be deployed and without with meeting those initial goals that I set out, that we really want to make this lightweight and of maximal utility to any industry partner that might want to deploy this. So we're kind of thinking about this in between these two extremes. Okay, so through that kind of thinking and through brainstorming there across the collaborative team and within my group at Rice, SeqScreen was born. Okay, and SeqScreen really just to kind of give you a high level, and I've presented this now in a few different ways. Today is going to be a slightly different way, and it's really through kind of this journey that I've been through with my collaborators over the last 5 years. So current classification tools, just and again, just in the DNA space specifically, misclassified near neighbor pathogens or ambiguous for emerging pathogens. That was something that really kind of inspired some other development choices of this tool. And then when we think about things in terms of functional annotation, what function is made this a sequence or sequence fragment carry? However, gene ontology annotations are complex, hierarchical, and vast, and not attuned to pathogenicity. So while a useful potential starting point, it cannot give us the fidelity of information or the level of information we may need to return an answer that we need to return. And then predictive modeling in terms of how do we predict what this might do. There's just not enough high quality training data available for functional mapping, and it's an imbalanced classification tasks. So really, after thinking about all of the challenges of this problem and mapping those out, did we finally come around to something that we, and again in collaboration with my collaborators at Signature Science, that we come up with a system that kind of really thought about sequences of concern or functions of sequences of concern, and using that as a framework to try to address some of the above listed challenges, but not fully solving them, of course, but really as a construct. And I'll get into those now. Okay, so I just said this phrase or term, functions of sequences of concern, or fun socks. Okay, what are fun socks? Well, not only are they fun looking socks, and you have an example on your slide, really we could think about them as biocurator defined annotations for proteins. Really, this again, the reason I've said this so many times, I really want to stress the fact that it's been 5 years of collaborative effort, and throughout those 5 years, the biocuration team, thanks to the literature that has been published, has developed a set of proteins that contain that are labeled as sequences of concern, potential concern, that contain certain terms that would indicate they could be harmful to human hosts. Now, again, nuance and context is everything, but this is again as a way of thinking about modeling this problem with limited training data and the limitations of the entire kind of goal of this kind of project. So as of this moment, we have 33 functions specific to sequences of concern. And in terms of training set, we had an initial set of 5,000 out of 30 million UTRF clusters, and we've expanded that out now to tens of thousands, but not much more than that. But again, given the lack of training data, it was on us to manually curate a set of training data, to which then we expanded on iteratively, which I'll kind of get to now. But I can pause here if there's any questions. If not, I'll just kind of keep going along. There's a question there. How do you make your training data? So there's a question that says, how do you make your training data sets? And that's a great question. So really, what it comes down to, it's really based off of kind of literature search through the team has done over the past several years, based on, of course, available resources. It's not just in a vacuum; it's through reading literature and with experts in virology and virulence and pathogenicity and microbial pathogenesis, and really compiling all of the existing literature and looking at essentially protein by protein and reading papers and saying, coming up with a mapping between, and I'll get to this again later, so I'll save the rest of the answer for later in the presentation, but coming up with a mapping of essentially protein to a set of these functions, one of these 33 functions. But a great question, and certainly a lot of effort, and it's something that we really look to the community to continue to engage with as well, because it's non-trivial and it may seem quite subjective, but I assure you, it's really kind of based off of annotation frameworks that exist. similar to if one were wanted to think about antibiotic resistance, et cetera. It's instead of just that category, we have 33 total. Okay. So I'll come back to that, the rest, to try to get the rest of your answer. But high level, okay, so really why do this, and why am I talking about fun socks? Well, it's really about moving beyond this idea of a bad microbe to sequence of concern. That's what inspired our approach, really to go beyond just saying, oh, we hit a list of 20 quote unquote bad microbes to which could be Bacillus anthracis or some other path, but just moving away from that model and say, what was it, given that we could be such a short fragment, we really need to think about which regions of the genome may indicate a sequence of concern or a function of sequence of concern. And as on the slide, there's a couple of references to papers that my team has published on this, but really the image is really just showing some examples of what we would consider functions of sequences of concern. So cytotoxicity, antibody neutralization, resist bacterial lysis, antimicrobial peptide degradation. So really thinking about what are all of the different things that could negatively impact a human host, for example. And this is just a small, quick vignette of that idea. Getting into this a little bit further, we have different categories. So maybe this would answer that question even further. Is okay. So we have damaging sequences of concern. We have direct acting sequences of concern. We have host cellular processes manipulated by sequences of concern, and we have immune subverting sequences of concern. So and then we have indirectly acting ones. So on this slide, you essentially we have these bins of sequences of concern, which you can think of as, again, these are all based off of previously published literature, where we're just kind of binning them out and populating them with example proteins. Some of this work has already been done, obviously, and some of it we're kind of dividing up bins into finer grain bins. But again, this is all based off of kind of prior work and then leveraging our team's expertise to refine further. But the whole point of all of this, getting back to the question, is really to create a confident set of training data for SeqScreen. We do all of this just so we can now train our machine learning algorithm on something. Okay, so and then this is actually the other part to the answer to that question is, now what do? We have a set of 5,000 proteins that were trained, that were created as training data, and now we essentially feed that into a machine learning algorithm that I'll describe that will predict additional proteins in the database that look like they may have additional functions of sequences of concern. And then we would take, so for example, let's just do a mock example here. Let's say round one of this, we would have 5,000, as I just described, that were developed over a year or two, feed that into a machine learning algorithm that now trains on that, tries to identify additional proteins that may have additional sequences of concern. It now returns a set of 15,000 from that 5,000. And then those extra 15,000 are then reviewed by our team. And then we iteratively went through this, a lot of work, but again, when you start really with no training data, you have to do this iteratively. We kind of refined everything, and we eventually saw that this iterative human in the loop process made the machine learning approach more accurate, and to the point where the biocreators were more, obviously more happy with or happier with the kind of the predictions it was making. Okay, and then just kind of high level, why didn't we just use something that existed? I mean, there was a lot of great resources out there, but specifically, we just thought that existing resources such as the virulence factor database just didn't encompass enough sequences of concern or functions of sequence of concern that we wanted to consider for the purposes of SeqScreen. For example, VFDB contained essentially the Go term overlap, the gene ontology term overlap with what we've created is not very much. It's only approximately 500 out of the 700 total Go terms, and so we encompass a much broader space than the kind of a comparable existing resource. So that's really the motivation for going through this. We learned that through the existing resources and the databases, they weren't broad enough to really accomplish the goal that we wanted to. Okay, I'm gonna shift gears a little bit. We're gonna get a little bit more into the technical details. I'm gonna go through this a little bit fast. But again, the paper is available on bioRxiv, and I'm available to answer questions. So please just interrupt me with questions if I just kind of don't make any of this clear. But essentially, high level, we'll try to go through this reasonably quick. Is again, now we have a set of, let's say, we have a set of 90,000 proteins, let's say. And this, where do we get this 90,000 from? Okay, so again, we want essentially to take that initial set of 5,000. We did that iterative loop, and we ended up with a set of 90,000 high confidence proteins that were of high quality annotation as indicated by Swissprot. Okay, and then we want to look at a set of features that would allow sufficient features for a machine learning algorithm to predict on new things. If a new thing were to come along, it would look at it and say, okay, we have sufficient information to make a prediction. So essentially, we have 90,000 proteins and 50,000 features, and then we essentially we took it down to essentially just dropping the rare features. We only want to keep the features that are common to at least 10 or more proteins, and that took us down to about 16,000 features. These features can be really anything specific to the protein that we can. I hear a little bit of background noise. Pause it. Yeah. Okay. And then yes, we have either, then the next step is either a feature selection method, which I'll just kind of breeze by, or we have a approach that can take that. So we can either have an approach that takes that information and then essentially picks a different machine learning method to do the FunSoc classification, or we can first go into a binary. Is this sequence likely to have FunSocs or not based on the features, and then do a classification. So either a kind of a first pass detection binary yes or no, and then classification, or just do the feature selection and classification right off the bat. Okay, so all of what I just described kind of maps onto this image that you have on your screen. Not that it's super important for the purposes of the talk, but just on the left-hand side, we trained on about 57% of our data that I just described. We validated on about 20%, and we tested on 25%. Okay, so and then again, we have a couple different approaches. We tried, we had to deal with class imbalance, et cetera, but we tried balance support vector classification plus a neural net here, in addition to a model 2, which is a multitask neural net, in addition to model 3, which is the multitask support vector classification. Okay, and this model 1 has 2 because it was really the binary, is it FunSoc containing yes or no, and then use the NN to then assign a FunSoc. And the FunSocs were one of these 33 categories that I described previously. So really we want to train and then on a new sequence and then assign it a FunSoc label. Okay, so all together it looks like this kind of large image where you just essentially feed in a short oligonucleotide sequence, run it through the machine learning classifier, and then it would run it. Seqscreen will run through all 3 of these, and then take the majority vote, and then assign the FunSocs, which could be there are no FunSocs, or there's 1 or 2 or 3 or 4, etc. And that's it'll do presence absence essentially for all of the 33 that we have, or 0. So it could be all zeros, or it could be a handful. Okay. How does this perform? Well, on that, the experimental setup I just described, essentially the ensemble, the bottom row, just kind of draw your attention to the bottom row of this table. If you use the ensemble method and you're looking at something like F1 score or precision or recall, macro precision or recall, or et cetera, we see that the machine learning method on short, and this is tested down to 50 to 100 nucleotide sequences and above, returns reasonably high precision and recall. Now, it isn't perfect, but we do see that the ensemble approach does help accuracy, but mostly precision, so accuracy or precision and sensitivity as well. So high level, the ensemble approach helped, and this took us a while to figure this out. And then if you line up all of the FunSocs in a single table, you ask it, because it really is a set of essentially 30. Seqscreen really is a set of 33 individual classifiers, one of which is antibiotic resistance, but more than that, there's 32 others. And if you look at that across the x-axis, the lines, the y-axis, if you get to the top, you're doing essentially the top precision or recall that you can get 100%, and then on the bottom of the y-axis is 0%. And if we just track with this, if we look at the red line, that's going to be the ensemble approach, and most of the time you'll see the red line tracking the best out of all of it. And the other point I'll say is there are some FunSocs on this image you'll see that drop down, and those when we looked into this, these are just FunSocs where we just have very few training examples in our data set. So these are things that we would benefit from additional training, training data to be able to improve the performance of those specific fund stocks. But overall, we see reasonably good performance across the variety of fund stocks. And this gave us confidence that now, given going back to the original kind of input that I described, given a DNA sequence input, we can now assign reasonably well to one of these 33 fund stocks. Okay. So high level, we'll back out now. That was the only technical deep dive we'll do on this talk. And again, I went through it kind of fast, so please feel free to reach out to me if you have any questions. But high level, there are 3 of the software. The main engine I just described, but it is a workflow that encompasses many components. And there's 3 different modes. One is kind of a fast, as we talked about at the beginning of this, as I talked about at the beginning of the talk, really about how it needs to be efficient. There's a fast mode which allows it to process large amounts of sequences and not too demanding of time. There's a sensitive mode for really applications that need to be maximally sensitive at the expense or at the cost of additional computational time. And then we're looking now to deploying this to log reads as well. So noisier, different input types. Okay, so with that, I got through my first lesson, essentially computational. So DNA, it's hard. Well, I still will kind of stand by that, but I feel like we have a reasonable framework for addressing that question, even though there's more work to do. This is and I think we've made quite a bit of good progress. I definitely am kind of attentively and acutely aware of all of the limitations here. Looks like I got a question here. How do you apply new data? Okay, so should I take questions now or save it? How are these normally ran? I'm happy to take them now or just wait to the end.
It's totally up to the speaker. If you're taking questions, please read the question first, because a lot of people will be watching the recording.
Okay. So so I may just continue then, and then just take them at the end. If that's okay, I will come back to your question. I'm sorry about not taking it right now. And then so lesson 2 is reference databases create bias, and high level. I published a paper back in 2018 that really looked at a lot of the tools that we have. I just described, like when we're matching sequences to a database, the results we get back are obviously biased by the composition of that database. And that's what this paper describes. And this is an open letter actually in Genome Biology that was published. So what do I mean by that? I'm going to give a short example. Essentially, in this example here, I'll just kind of shoot ahead. If you were to take DNA sequence that was simulated from Clostridium sporogenes or Clostridium botulinum, and so you have 2 genomes, 2 bacterial genomes that are very closely related, and you simulate reads or sequences from both, and then you run it through a variety of classification methods, which you see in this top table. Right? It's several listed there. Each column is a different method after the column true organism. And we see that sometimes each method will sometimes, like if the true organism is C. sporogenes, some methods will get that correct, and some will call it Clostridium botulinum. Why is that important? Well, one, Clostridium botulinum has an SC toxin, and we want to be able to classify it accurately as Clostridium botulinum if it is that, for example, and we don't want to falsely classify it. So we see on the top row that some tools falsely classify as a pathogen being present, CBOT, and on the bottom is the exact opposite. Some tools will get that right, but then it will falsely classify it as the near neighbor. And why does this happen? So very high level in this paper, we kind of describe this, but essentially you have genomes, you have a biased representation of pathogens in the database, and that will affect the performance of different computational tools that perform taxonomic classification, high level. Okay, if you would like to know more about that in the confines of this talk, I don't have time to fully dive into this paper, but we get into this, and there's been subsequent papers that have dug further into this question. Okay, so near neighbor wise, but we go back to SeqScreen here, we have on the screen, and essentially SeqScreen is able to, since it focuses on functions of sequences of concern, it really doesn't care if it's CBOT or C. sporogenes. What it really cares about, if it doesn't find that neurotoxin. And in fact, it does. So it reports there's a neurotoxin here. I don't really care what the taxonomic classification says, but I do find a fun sock, a protein containing fun socks, and it returns it, and that's what you see in your screen. Okay, and then lesson number 3, context is everything. And again, I'm sorry, I don't have for the limits of the talk. I think I have maybe 10 more minutes here. I'm going to go through it reasonably fast. But I'll summarize all of these at the end, by the way. Context is everything. We published a paper in PLOS Pathogens Pearl that really got into the nuances of predicting pathogenicity and the impetus for novel computational approaches. This is in collaboration with a former postdoctoral scientist in my group, Leo Elworth, and then also signature science scientist, Dr. Krista Turnus. And so we wrote this really thinking about how does context affect everything I just told you. Again, I don't have the time to really get into it. So high level, let me just describe a high level example here. And this is something that happened to me. And this is just to tell everyone, AI or ML machine learning isn't perfect. And I'm sure everyone knows this, but just a fun example I'll walk everyone through. So I was trying to access some pages that required a reCAPTCHA. And that's the screen you see. In the screen you see, you'll see 9 squares. And it says select all images with parking meters. And I really wanted to get to the next page to finish doing something I needed to log in or pay for something, right? And it said, please select all images with parking meters. And I checked the 2 parking meters that seemed like parking meters to me, but it would not let me advance. And I said, why is this? And I looked and I said, well, it must have a corrupted truth set that thinks that the mailbox in the middle of a grass field is a parking meter. And so once I selected that as a parking meter and hit next, it let me advance. But so obviously something was wrong in the truth data set. Now, similarly to the problem that SeekScreen looks at, you really need to consider context. Now, even if that were a parking meter, why, the question would be, why is this sitting in the middle of a grass field with no parking spot around it? So really, this is a reasonably good analogy for how we need to think about sequences of concern. What is nearby? What other sequences of concern do we see when we're performing this computational analysis? Okay, and so context is key. Pathogen, host, and environment. Really need to keep those into consideration. Yep. Yeah, exactly. The mailbox is a parking meter, but unfortunately it isn't, and it frustrated me. I was like, "Wow, look!" But really, context is key. Okay, so getting back into the paper. So a glimpse again. This is from the paper I just shared. Is really what makes this problem kind of challenging. Is when we get more into the unknown. So from the x-axis, you're going from less difficult to more difficult, and the y-axis, you're going from low pathogenicity to high pathogenicity. So what really kind of gets hard is when you're going from known to unknown, right? This is an open challenge when you're looking at things that are kind of known to unknown. So anything, if we get into the novel virulence factors, unknown housekeeping genes, these are things that can really confound existing approaches, and something that we really need to continue to work towards. Now, there's been some recent approaches specific to deep learning, et cetera, which may be able to help us out in these spaces, but really thinking about this not only context wise, but also novelty is a really big confounder as well. And then two more lessons, then I'll come back to the questions. And when I wrap up, is I really want to say community engagement is key. This is another big lesson I've learned over the last 5 years. Not only have I depended on or relied on community feedback, input, guidance as I work through these questions, it's really exciting to see the active amount of community engagement going on, such as this workshop report put out by Sarah Carter, Beth Vitalis, et cetera, talking about genome engineering and biosecurity, industry practices, resources, and opportunities. So really, this is just one example of a recent activity, but really a recent call, to June 28th this year. Essentially, there's governments requesting feedback for a screening framework on guidance for providers of synthetic double-stranded DNA. So really, this is not only has it been a community, it learned so much from community engagement. Right now we have time to work together and help refine and advise on guidance specific to DNA screening. So community is key, and engagement is key, and that's something I've really benefited from engagement with EBRC and many other stakeholders in the community. And then just lesson number 5. I will end on this, essentially taking this back to where I started, metagenomics, metagenomic classification, metagenomic analysis. Really after working on this problem for many years, realized that the care we've taken to kind of build these tools out really apply back to just if you want to look at a metagenomic sample or a microbiome. And this is a vignette that will highlight this. Essentially, we wanted to look at a case and control sample where there was a control sample of a patient that did not have COVID versus a COVID patient case that a previous study had looked at, did not find any SARS-CoV-2 presence despite the COVID positive test, and it was a pretty severe kind of outcome, and it could have just been a limitation and or from when the sample was sequenced or et cetera. But we wanted to take a look at this data set to see if SeqScreen could look at this at all and add any additional information to it. Okay, and really what we're going to look at is gene ontology terms tied to a funsock specific to suppression of virus by virus of host, induction by virus of host mRNA, and suppression by virus of host NF-kappa B. So we're really looking for the funsocks that have these gene ontology terms. So we ran it on this meta-transcriptomic data set. And so kind of as a punchline coming back to this, after we went through and ran it on, millions of short sequence reads, which wasn't the original design of what we intended for SeqScreen, but it actually comes back, since we kept it efficient and accurate and kind of detailed, we actually can now run it on these data sets. And it came back and told us that we're just a handful of reads inside of that sample that belong to SARS-CoV-2. And I want to note that when we did this experiment, we intentionally removed all SARS-CoV-2 genomes from the database as well as near neighbor sequences, and it still was able to find that there were reads that mapped to functions of sequences of concern, and turns out the reads that it found are listed on the bottom of the slide matched perfectly to the SARS-CoV-2 genome. So again, this is just another way of viewing this. When I talked about there and back again, really I started this thinking about metagenomic classification. I kind of really then focused on the specific problem of synthetic DNA screening. And now I'm back to saying these tools, not only are they useful for the purposes of DNA screening, these tools can really provide additional information to screening environmental samples that may contain pathogens. Okay. So summarizing all my lessons as my time is kind of wrapping up here, I really want to say, again, emphasize these 5 lessons over the last 5 years. Kind of lesson number 1, the fact that DNA sequence screening is hard, and I will stand by that statement. The lesson number 2 I present today is the reference databases create bias. Lesson number 3, context is everything. Lesson number 4, community engagement is key. And 5, this idea that DNA screening approaches can help feed back into improving metagenomic classification. It's kind of a unexpected end to where we're at currently. And of course, there's additional work that can be done, but it really comes back to this idea of the bias in the database and really thinking it more of a function of sequences of concern perspective. And again, the last thing I want to say is that SeqScreen is complementary to many of the existing approaches. I'm not saying to stop using some these other approaches, which have performed really, really well. I'm saying for cases where things are exceptionally challenging, where there might be some novel virulence factor, or there might be some ambiguity, this is another tool that biologists can use, or et cetera, when they want additional information from their samples. Okay. And with that, I will just kind of have a short plug for the software here. This is just obviously that I've said this a few times now, but there's the URL where you can access it. It's open source, free to use. Community feedback is really welcome. And then I just want to acknowledge the entire team and the source of funding here, especially the lab members here that invite Balaji, who's been really leading the charge on the software development side, and then of course Signature Science, that has played such a critical role in the development of this platform. And with that, I will stop my share, and I'm happy to take any questions, and I'll start with the questions I got before.
Thank you so much. Yeah, if you could read a question before you answer, that's going to help people watching.
All right, thank you. So I'm going to go back to my one question here. It says, this is very important and interesting. I may miss it, but how to apply new dangerous toxin sequences of viruses that do not have sequence history or training? Also, how to identify hybrid synthetic sequence. This is wonderful. Well, thank you, Tae-Suk. So I will say, the first question I would say, for new toxins or viruses, history of training. Now, that's a nuanced point, but we've actually done this to our, we've looked into this where we'll remove, so for the BOD-A neurotoxin example. We essentially ran it through the BOD-A genome, and we took out all BOD-A proteins, anything within I think 20 or 30% amino acid similarity, just wiped it all away from the database. And there were still a distant homologue in the database that allowed us to classify this. Now, so your question, well, it's a really good question. The only time we've had an issue there is when you really just say there's some novel thing that has no similar protein within 50% similarity. Now, in that case, I'm not sure what can be done. I think there's some ideas, but certainly in that use case, seq is not going to do a good job. But if you're anywhere in that space, it will return an answer, and more times than not, in our hands, it's actually going to do a pretty good job with that. So, and then number two, hybrid. Hybrid is a great second segue. That's actually for the long read analysis. You actually have to handle hybrid, where half of the sequence could be some fun stock containing thing, and the other half could be something completely different. So in that development efforts, we've now started to look into splitting things out and interpreting multiple hits across the sequence length, and how do we interpret that? And this could be even thinking about, oh, is this a chimeric sequence, or what? But we now report this out in different ways. So the current version will go ahead and split the sequence up, and that was a non-trivial task, because we're not doing ORF, or we're not doing gene calling on the read. So you really have to identify the boundaries of the hits accurately, and we report those out. So step one is kind of complete, but step two, maybe where your question is hinting to more, is how do you interpret that? I think that there are some things from this IARPA program Felix that we might be able to learn from, but this idea of detecting different types of engineering events, I think will help characterize kind of what we're doing. But right now we can now handle that. Previously we could not handle it.
So that's fantastic. One follow-up question. So in that case, I mean, if we have another new virus, I mean, we never seen before, we could kind of identify that as sort of, deadly virus. Can we do that, right?
Yeah,
Wonderful.
yeah, and and again, I'm going to put some nuance there. Of course, there could be, but a lot of what we've done in terms of our experiments, or even things that have recently emerged, it will come up as, say, there's something here of concern. You may, and then you take a look, and again, this is why I think the moving away from the taxonomic approach, while it's useful, it's so important, because it just may either misclassify or be, as you know, viruses that are just so different at the sequence level that it might not be able to say anything. So it's a great point. Thanks for that question.
Thank you so much. This is so exciting. So thank you so much, and I'm looking forward to your next paper. So I actually need to deep, but that's fantastic.
Yep. Thanks. So I got a question. This is, what is the codon and mutation tolerance of the screening algorithm? Would sequence of concern be flagged if it is 1, 2, or more AA different? Okay, so it has high tolerance. We've tested a lot, so it can be quite different. So in terms of sensitivity, it's quite high. But in terms of, can we detect? So if there's a sequence of concern that's one mutation away from a sequence that's not mutation sequence of concern, could we detect that? Well, I would say probably one mutation away, maybe not. We do have a module inside of the software that's called outlier detection. This is with our collaborators at University of Maryland, Dr. Mihai Pop there, where when it's looking at the multiple sequence alignment, we've seen it split off sequences with as few as 3 or 4 or 5 mutations away. But I think one might, it's unclear, we'd have to look at that specific case, but we have thought about how do you actually get down to individual sequences, mutation that separate out different sequences of concern. Great question. I would say that's more of an open research area, but we have made some progress in that area. Okay, and then it says, there's a question that says, do you work with any DNA synthesis companies to implement this algorithm? We didn't work with any DNA synthesis companies to implement the algorithm, but we do have company partners and collaborators that have given us advice and feedback as we've developed it. And so so in some sense, we've gotten their feedback, we've gotten their time and guidance, but the implementation of the algorithm was strictly across the team that I kind of described. And then and then so what is the most common application from Peter? Preventing synthesis errors, or is it more aimed at preventing bad actors? That's a great question. I don't really have a great insight, like common application. I think if I were to say of that, my guess is more about synthesis errors. I'm not behind the scenes there. I don't really know. And honestly, I really think about this tool more towards how do we get better at pathogen identification that Tae-Suk had mentioned. What if we, how do we really get better at tracking novel emerging pathogens? And I see this as a way towards that. Now, for, again, the DNA synthesis screening, yeah, you could think of a variety of applications. One could just be saying, we want to avoid errors, and, help them that way. But the bad actor application, obviously it's one of the motivating factors behind the prior research as well. But again, I think, to me personally, just speaking very freely here, I think one of the most exciting applications is kind of the going off into detection of novel emerging pathogens. And then the last one is, what do you think of the new U.S. federal screening guidance proposal? Is it improved? Great question. I'm still discussing this with many collaborators and partners. I think there has parts of it. I think the language is improved. I think there are some nuance there that still needs to be discussed and worked out. And I again, this is where, going back to my point in terms of lessons learned, this is really where community engagement is key. I can have my own thoughts, but I'm not sure. I really need to engage with others who have better expertise in the areas of either kind of thinking about this and implications across the industry and the government to really comment on that with any level of confidence. So again, it's kind of a deferred question, but really with the intention of saying, this is where I really rely on engagement from others to kind of discuss this and say, what do you all think of this language? Here's my perspective, and then coming together to say, here's an idea for improved. But overall, just high level, it looks like they have made improvements in certain areas. But again, I think I'd like to discuss further with others who have thought about this as well. With that, I think I got through all of the questions, unless there's any more.
I want to say thank you very much. I had a couple of questions, but they were asked by others in chat. I'm especially interested in the mutation screening, but you answered that. Excellent. So thank you very much for the talk. Thank you, everyone, for the discussion. And enjoy your evening. Enjoy the rest of your evening.
Yeah, will do. Thank you. Thanks again, Kate, for the invitation. It was great to be here to talk with you all. And again, I'm happy to follow up. I know I went through some of the material pretty fast, so happy to follow up with anyone if there's any additional questions. And yeah, thank you.
Thank you. Bye.
Bye.